Hello everyone, I am Prateek and I am a plant scientist. Today I will walk you through to our exciting research in which I am involved in towards our efforts to answer the serious question of world hunger. Do you know that every year we lose trillions of dollars due to reduced crop productivity, which is an outcome of unfavorable human activities and global climate change? So, to make our crops better and improved in response to these aberrant changes, we are trying to understand the circadian clock regulation of plant growth and development. Yes, you heard it right. Plants do have the internal clock which governs their growth and development like us humans. They do sense seasonal variations, get hungry and sick, and at the same time try to adapt to cope up with these conditions. Before we go to focus on our research question, which is how plants maintains the clock and uses this information as a sensing mechanism to govern and fine-tune the growth and developmental machinery. For example, how they decide to flower and when to flower. Let's talk a little bit about the structure of the clock. In a simplified view, the clock structure comprises of three core components. First is input, which is how plants keep and set the time in response to the local environment. Second is an oscillator, which is how plants keep the track of the passing time. For example, like a calendar. And third is an output, which is how plants uses the knowledge of the clock to adjust their daily activities. However, this version is not as simple as it seems. Different genetic and environmental factors makes the clock complicated, interconnected wiring system. So it is really important to untangle this wiring system layer by layer to uncover the genetic base, but at the same time, it is equally important to consider the environmental factors which can reset the clock and mess the system completely. In our lab, to study the clock, we use model plant Arabidopsis. Why Arabidopsis? I mean, it's a wheat. It can be found growing everywhere and anywhere. But it has been proven as a sunshine resource scientifically because it's an annual flowering plant with a relatively short life cycle and a small genome. Over the years, findings from the Arabidopsis study have proven to be very instrumental in converging the knowledge to the fields towards making better crops in response to these changes. We are trying to identify and understand a role of a gene called BBOX32 or commonly known as BBX32. A gene is a piece of DNA that contains coded information of an organism and passes this information from one generation to another. It has been shown that BBX32 has a huge impact on crop productivity. When the soybean plants with higher level of BBX32 were grown in fields, the, it increased the broad acre yield. And that makes us wonder how one single gene could be that impactful. And hence we decided to unravel the molecular mechanism of BBX32 and its mode of action in Arabidopsis. What we found was BBX32 was under the direct control of the clock, and it governs the flowering time regulation in plant. In fact, our study defines how this gene exerts an effect on crop growth and yield, which could be a profitable strategy to develop early and late flowering plants. Early flowering plant could be advantageous for the crops cultivated primarily for seeds, while late flowering could be an humongous advantageous when the total biomass is the goal of the production. Thus, it is really very important to save our planet and make it more compatible with healthy and green plants and ultimately food for all. Plants don't cry. That doesn't mean that they don't get hurt. They do. But when they make us realize that, it is usually too late. Think about it. If we have food on our table and for our kids every day, despite the political situations, we all will be very happy and less stressful. With this, I would like to thank my mentor, Dr. Steve K, for giving me this amazing opportunity and training. Also, my Art of Science Communication mentors, Deborah and Greg, and the group members, Veronica, Jessica, and Pushpanathan, for their valuable comments and suggestions in my effort to be effective and a good science communicator. And last but not the least, Jeff and ASBMB for organizing and putting together this amazing course for all of us. So thank you all, and I wish all the participants good luck. Bye-bye.